Okay, so this is part 2 of the transcription lecture, and this is where we left off on from part 1. And so remember, I told you that you guys are really just responsible for the details for negative control of that lac operon. This is just like I said, I sometimes will give the detailed explanation about the positive control of the lac operon, which is basically due to the presence of glucose um, or the absence of glucose, in that there is a regulatory protein that will actually bind to that, to essentially the promoter of that lac operon and turn everything on. Okay, and so if the regulatory protein binds and activates transcription or turns things on, then it's called positive control. Okay, but you don't need to know those details. So now we're going to move on to talk about eukaryotic RNA polymerases and also general transcription factors. So basically, we're going to talk about initiation of transcription in eukaryotes. We already talked about it in prokaryotes. And actually, so that I don't forget, if you look at and remember, again, we're filling in our review sheet here, for chapter 8. Okay, so we talked about negative control of the lac operon, and now we're talking about eukaryotic general transcription factors and basically initiation of transcription. If you look back at your chapter 8 homework questions, the first question asks, what is the role of sigma factors, right, or the sigma subunit in bacterial RNA synthesis? And so we talked about this, so let's just go back and do this now so I don't have to come all the way back to this. Again, we're talking about initiation of transcription, and remember that the E. coli RNA polymerase is really a large protein, right, with multiple subunits, okay? And so the question is asking you about the sigma subunits. Remember, that's the subunit that actually will recognize the sequences, right, particularly these -10 and -35 sequences in the promoter, right? And then once that binds, basically the other subunits sort of are there to start functioning, but really the function of the sigma subunit is to bind to the promoter and really to identify where transcription is going to start, right? And then basically transcription will start, right? And then after about 10 nucleotides or so, that sigma subunit will fall off. Okay, so the function of the sigma factors, or the sigma subunit, are really to recognize the promoter, right, and initiate transcription, and then from there they'll be removed. Okay, so again, this is all in the beginning here of the lecture. Okay, so let's go back to eukaryotes, and then I'll make sure I answer those other 2 questions from chapter 8 as we go through. Okay. So instead of just one RNA polymerase in prokaryotes, like we just saw, in eukaryotes there are 3 RNA polymerases, okay, and they will transcribe different classes of genes. And so okay, and so if you guys flip to page 3 of your review sheet, you'll see it says eukaryotic RNA polymerases. Okay, so there's a table here that's going to show you some of this information, table 8.1. So you want to make sure you write down those different types of RNA polymerases. Okay. All right. So it says RNA polymerases also must interact with additional proteins to initiate and regulate transcription. So it's more complex in a eukaryote than prokaryote. Prokaryote is really just that one RNA polymerase with those multiple subunits. All of those together is enough to initiate and to start transcription. Okay. But however, in eukaryotes, it's more complicated. Transcription takes place on chromatin, okay? And so there needs to be some regulation of chromatin structure, which will also be important in regulating gene expression. Okay, so eukaryotic RNA polymerase is about 12 to 17 different subunits. So again, think about this compared to what we saw with the RNA polymerases. They have 9 conserved subunits, meaning 9 subunits that are the same across all of those RNA polymerases, and 5 out of those 9 are closely related to what we talked about with the E. coli RNA polymerase. Okay, and the yeast RNA polymerase, which yes is a eukaryote, is actually similar. One specific yeast RNA polymerase 2, is very similar to the bacteria or E. coli RNA polymerase. All right, so this is that table that I was just referring to, and again, it's on your review sheet on the top of page 3. You can see in terms of the type of RNA that gets transcribed. Okay, so RNA polymerase II does messenger RNA. So that's our main RNA that we talk about most. We need to know that. We need to know that RNA polymerase II will transcribe genes that are going to code for, or you're going to make mRNA. Okay. Remember we talked about different types of RNAs before in our other lecture. I guess that was in chapter 6, right? And you can actually go back to remind yourself about some of these if you even just look back on page 1 of your review sheet, right? We talked about some microRNAs, long non-coding RNAs. Those are actually also transcribed by RNA polymerase II as well. We talk about transfer RNA, it's RNA polymerase III, okay? Ribosomal RNA, it depends on what you're talking about. So both RNA polymerase 1 and 3, okay? And we'll talk more about ribosomal RNA when we get to the next chapter on protein processing, okay? But you should know this. You don't need to worry about the snRNA and the scRNA, but from basically here down, you guys need to know which RNA polymerase basically is responsible for transcription of what. So RNA polymerase II for mRNA, all right, for tRNA, it's RNA polymerase III, okay? And we should know for rRNA, it's both 1 and 3, depending on which type of rRNA you're talking about. Okay, this is actually showing you this structure of yeast RNA polymerase II, and so you can see. Okay, sorry, just to continue on here. So this is just showing you the structure of the yeast RNA polymerase II, and you can see it's similar structure to the bacterial RNA polymerase that we talked about in the beginning of chapter. Each different color here represents a different subunit. And you can see there's more than there were in the E. coli, okay? All right, so we said RNA polymerase II. Again, you should definitely remember this, that RNA polymerase II synthesizes mRNA. Okay, and so obviously you focus most on this because we're talking about the production of messenger RNA. There are many initiation factors that are needed, okay, not just those sigma factors like in bacteria, okay, to basically initiate this. And they're actually not even associated with the polymerase, so they're different proteins that are needed. Okay, so we call these general transcription factors. And so again, if you look at your review sheet, general transcription factors, mediator complex, we're going to have some details around that. Okay, so if we talk about initiation of transcription by RNA polymerase II, which is going to make mRNA, right? These are all the transcription factors, all the proteins that are needed to initiate that. Okay, so general transcription factors are proteins that are needed to initiate transcription of or initiate eukaryotic transcription. Here we're specifically going to talk about RNA polymerase II, right, and the initiation of transcription in eukaryotes, right, and the production of mRNA. Okay, it says about 10% of the genes in the human genome actually encode transcription factors, right? So there is a large amount of the genome that is actually dedicated to encoding transcription factors. All right, so just like we talked about a promoter with the E. coli, okay, there are several sequence elements that surround that transcription site. So before we talked about the -10 and the -35 region, right, here we have some different terminology. So the TATA box, so TATA, that's the sequence that's within that region, actually resembles that -10 sequence that we saw in that bacterial promoter, okay? There are others as well, okay? So other regions of that promoter, remember the promoter sequence is on the DNA, right? So things like LNR elements, transcription factor 2B recognition elements, BRE, okay, DCE, these are all elements that are part of the promoter. So you can see that even the promoter is more complex than the E. coli. All right, in terms of the general transcription factors that are required for initiation, okay, here they are listed. Transcription factor 2D, okay, has multiple subunits, including a subunit that's going to bind to that TATA box of the promoter, right? This TBP. There are other subunits that will bind these other sequences that I mentioned, okay? Again, remember all of these sequences are part of the DNA. These are part of the promoter, okay? More complex. This transcription factor 2D is actually a protein that's going to bind to these sequences here, okay? There are other transcription factors as well. Transcription factor 2B, 2F, 2E, and 2H will all bind in association with that RNA polymerase to basically form this pre-initiation complex. And then you'll see there's going to be another protein that has to come in before transcription can actually start. Okay, so here's your transcription start site. Remember we talked about upstream versus downstream. These are parts of the promoter that are upstream of the start site. These are kind of right around the start site, a little bit downstream. These are all part of the promoter complex. These are all just regions of the DNA to which parts of this sort of huge complex, right, huge, large transcription factor is going to bind to. So, right, so parts of this all bind to these different parts of these elements in the promoter sequence, and that's what we talked about here, okay? So you can kind of see this big blob here, this TF2B, TF2D first, right? Binding essentially at some point to all of these sequences that we mentioned, and then TF2B will come in. That's in green here, okay? So this is TF2D, and this is TF2B. So the first one that binds is this TF2D, and then TF2B. Then the RNA polymerase will bind along with this general transcription factor TF2F. Okay, so you can kind of see they sort of bind in a sequence. Okay, this is basically showing you this C-terminal domain. I'll mention that again in a second. Okay, it's just the C-terminal domain, basically part of the RNA polymerase. Okay, so it's not the mRNA being formed or anything like that. So that's why I write this in here, because it's a little misleading. All right, and so now you can see some more of these general transcription factors can't come in to bind. So for instance, if this transcription factor 2F doesn't bind, then E and H won't be able to come in. So it's important that all of these are bound and bind in sequence, and all of them have separate independent functions, okay? All right, now, like I told you, this is this pre-initiation complex, okay? And so, all right, so I kind of have it lumped together in your review sheet, but basically these are all proteins or general transcription factors that are part of the pre-initiation complex. So transcription is still not ready to start, okay? There's one more thing that has to come in, and basically this is a protein called the mediator, which is actually a protein that has over 20 subunits, okay? So it's a huge protein, and it will interact with both the general transcription factors and RNA polymerase. So you can kind of think of it as sort of coming in to make sure everything is there, right? Ready to go in the right position, and then transcription will start, okay? Actually, there's sort of a change that occurs to sort of initiate it. So mediator comes in, and when that mediator protein comes and binds, it's sort of whatever the signal that kind of allows this RNA polymerase, or the change that allows this RNA polymerase to separate from the rest of this complex and actually start transcription, is actually phosphorylation of the C-terminal domain. So that's why we have this sort of, pointed out here, okay? And so again, this mediator binds, cause phosphorylation of the C-terminal domain of the RNA polymerase, which allows it to separate from the rest of this complex. And then you can see now RNA is being produced and transcription has started. Okay, so all of what we just talked about was initiation of transcription in eukaryotes. Okay, it involves a multitude of general transcription factors, right, that all bind in sequence, including RNA polymerase 2, because we're talking about the synthesis of mRNA, messenger RNA. Then you have that mediator, that large mediator protein that will come along. The RNA polymerase, the C-terminal domain of the RNA polymerase becomes phosphorylated, and then it separates and transcription will start. Okay? All right. Now, I don't hold you guys really responsible for the details of this, but I like to put this in just so you guys can see that the transcription, or the initiation of transcription, right, in general transcription factor with other polymerases, right, like RNA polymerase 1 that transcribes some ribosomal RNA genes, it's different. Okay, there's different proteins involved. And so I have just written on here transcription by RNA polymerase 1, ribosomal RNA genes. So know the basics of this one. We're not going to go into details with all the rest. And so it says here, right, if you're going to transcribe these ribosomal RNA genes, basically it's making, it's going to make these sort of pre-ribosomal RNAs, which then get processed to yield these ribosomal RNAs. And then eventually, right, it's going to be part of the ribosome, okay? And so these are just different parts of or different types of ribosomal RNA, okay? And again, we're going to talk more about this when we get to the protein part, so that's why I don't like to spend too much time on it right now. So the promoters of the ribosomal rRNA genes are different than what we just talked about, okay? So again, I want you to understand that there are differences here. And they're recognized by different transcription factors than what we talked about with RNA polymerase chain, okay? And so some of these factors are UBF, SL1. So these are different proteins, okay? So you can see here, right, here's the promoter, just like we talked about before. These are ribosomal RNA genes, okay? It's polymerase 1 that's going to bind, not polymerase 2. And you can see that the proteins or the transcription factors, the general transcription factors are different, right? And so, and again, overall, you can see this is a little bit more simple process than what we talked about before. So again, if you're going to talk about other polymerases, because remember I showed you that table and I said we should know the different polymerases and what they transcribe, right? Transfer RNA, okay? Also the 5S RNA are transcribed by RNA polymerase 3, like I showed you in that table, okay? And there are 3 different types of promoters and different transcription factors that will bind here, okay? So you can see different transcription factors depending on which one we're talking about. And again, the specifics are different. You don't need to know these specifics, okay? I'm not going to ask you which transcription factors bind, you know, the 5S, or which transcription factors are involved in initiating the 5S ribosomal RNA versus tRNA, okay? We just need to know what polymerase is involved. So RNA polymerase 3, right, for this 5S ribosomal RNA, tRNA, and I told you not to worry about the small nuclear RNA. And know that if we go back here, right, for ribosomal, the other ribosomal RNA types, that's polymerase 1. And for mRNA, I'm just reviewing, which polymerase is it? Hopefully you can kind of think of it. It's RNA polymerase 2. Okay. All right, so that was all general transcription factors and initiation of transcription. Okay, remember that transcription, or the cell, does not transcribe its entire genome, right? It only transcribes those genes that it needs to make at that given point in time. And so it's all initiated by the binding of these general transcription factors. Okay, so these are just, again, general transcription factors that allow transcription to happen, okay? Because we are going to talk about other specific transcription factors that will regulate this process, okay? So if you talk about general transcription factors, you're just thinking about what we just went over in terms of those proteins that are involved in just initiating transcription pretty much all the time, right? So no matter what genes you're talking about, you're going to have these general transcription factors involved. Like I said, then we will talk about more specific sort of types of proteins that will regulate, okay? So we haven't really been talking, none of what we've talked about so far is really regulation. The only thing that we talked about that involves regulation was with bacteria and talking about the lac operon, right? We talked about that's the way that bacteria or prokaryotes sort of regulate transcription. We haven't talked about anything in terms of eukaryotes yet, okay? Okay, so now we're going to start talking about regulation in eukaryotes, okay? And again, you can see that on your review sheet, regulation of transcription in eukaryotes. And so we have it sort of categorized the way it's listed on this review sheet. Cis-acting regulatory sequences, right? Transcriptional regulatory proteins, okay? All right. And you can see there's a few things that are listed that say they're not on the exam. So these specific sort of techniques, this gene transfer assay and the DNA affinity chromatography, I'm not going to test you on the exam. And so I may sort of skip over that kind of quickly. Okay. All right, so it says eukaryotic DNA is packaged into chromatin. We've talked about this before, and so that will limit its availability for transcription. Even remember, we talked about the difference between euchromatin and heterochromatin, and we say, you know, the difference was one's more transcriptionally active than the other. Okay. Okay, so a little bit more kind of on that sort of idea here, okay? So non-coding RNAs and proteins will regulate transcription via modifications of chromatin structure, right? Meaning, if there's a part of the chromatin that's really tightly wound, you have to, if you want to transcribe those genes, right, that structure has to be modified to make the DNA available. There are many sequences, cis-acting sequences, meaning, right, these are nearby the sort of genes that you're going to transcribe, that will regulate expression. And the assays that were able to identify these regulatory sequences were called these gene transfer assays. And I told you don't really need to know about the details and how these gene transfer assays work, okay? But basically, you can ligate these regulatory sequences, right, to reporter genes, meaning genes that will allow you to see, right, to see them or to detect that they're there, or that they've been transcribed, essentially. And then that regulatory sequence will direct the expression of that reporter gene. Okay? And so you can kind of see this here, right? So this regulatory sequence is basically going to direct the transcription of this reporter gene, and again, meaning it's a gene that you can see, right? So if you transfect some cells, right, this plasmid ends up inside the cells, right? They're going to basically transcribe this reporter gene, and then you're going to be able to see that, okay? And so that's one of the ways that they were able to identify these regulatory sequences, right? So I'm not going to ask you about the details. Okay, so a type of regulatory sequence that's located farther away from the start site, right? So this is kind of saying cis-acting, meaning, right, that regulatory sequence is right in front. So almost think of it as, you know, as that promoter that we showed before, right? When we talked about just initiation of general transcription, the regulatory sequence sits right in front of that gene, okay? All right. So we're gonna talk more about the promoter in a second, okay? Now, and again, remember, sorry, let's go back here because I think I just kind of misspoke. The cis-acting means that it's on the, you know, the same piece of DNA. What? Oh, now I just, is it that it's, yeah. So anyway, we're gonna continue with this, but I'll make sure I clarify that in a second. Okay. All right. So a type of regulatory sequence, right? And again, it's under cis-acting here, is a enhancer. Okay, so these can be located farther away, right? And so these were first identified using a viral promoter, this SV40, and basically the activity of the enhancer doesn't depend on their distance from the transcription initiation site or even how they're inserted in, right? So if you look, this is showing you, so here's an example, this enhancer is right in front of the promoter, okay? So meaning if something binds to this enhancer, it's going to increase the amount of transcription that occurs, you know, downstream of this promoter, okay? But you can have this enhancer basically inserted anywhere. So if you look here and look at the action of an enhancer, right? Here's your promoter, here's your gene, okay? And that's sort of just basally or sort of normal levels of transcription, how much mRNA is made from that gene, right? If an enhancer is there, you can see it stimulates the transcription. Again, enhancer, enhance, stimulates it. If it's really far upstream, it will still have the same effect. If it's downstream, it will still have the same effect. If it's actually inserted in the reverse direction, it will still have the same effect, okay? All right, so there you go. Enhancers, it says like promoters will bind transcription factor. That's okay. So like I said, again, an enhancer, here's your promoter and here's your enhancer. These are both places in the DNA, right? Or sequences of DNA to which things will bind to regulate it, okay? So that's what this is telling you here. So it says DNA looping will allow a transcription factor bound to a distant enhancer to interact with those proteins associated with that RNA polymerase mediator complex. So remember, I told you all those general transcription factors are there no matter what gene you're transcribing, right? But then these other regulatory proteins will interact with them to either increase, enhance, like in the case of an enhancer, increase transcription, or they can also decrease transcription. Okay, so this is showing you here because the idea, and you don't need to know these specifics in terms of these different proteins, but think about it. if this enhancer is so far away, how does it actually interact with what's going on here? And the answer is that it can actually form a loop and actually become in contact with that promoter essentially and the initiation start site of transcription. Okay, so that's what this is showing you. So this DNA looping is involved when an enhancer is really far away from the initiation, transcription initiation start site. Okay. So here's an example of an enhancer. So an enhancer controls transcription of immunoglobulin genes in B lymphocytes. Okay, so it shows that this enhancer is active in lymphocytes, but not in other cell types. Okay, and so it's partly that regulatory sequence, that enhancer sequence, is partly responsible for the specific expression of immunoglobulin genes, right? And so let's look at a figure of this in a second, right? So this immunoglobulin heavy chain enhancer has at least 9 sequence elements that are protein binding sites. Okay, so you can kind of see how this is super important. Okay, if we're talking about, B cells, they make antibodies. Okay, and so here, right, and this is, I guess, a more complex example, but I just wanted to give you guys an example. You can see, all of these are different. These are different parts of the enhancer, right? So from kind of each part here, a different regulatory protein combined, and depending on which proteins are bound to these regulatory sequences, it's going to affect the transcription of those immunoglobulin genes. Okay, and so you can see that sort of different combinations of different proteins bound will affect how that transcription happens. Okay, so this is definitely a complex example of an enhancer. All right, so again, more for about 10% of the human genome. Okay, super important these enhancers are. If you have mutations in the enhancers, they will, not affect the protein coding sequence, but they're going to affect the activity of that protein essentially, right? And how much is made. And so if you don't make enough of a certain enzyme, or you don't make enough of a certain, peptide hormone, that's a problem, right? All right, and so it says in any given cell too that there are multiple enhancers that will work together to regulate individual genes. So it is very complex. It's not like there's just one enhancer that will sort of control one gene, right? Just like you saw with that immunoglobulin example, that's really complex, right? Many different proteins combine there to regulate that, okay? So again, take home message, bottom line here is an enhancer is a sequence, right, of the DNA that can really be located anywhere with respect to the transcription start site, okay? And they enhance the activity or they enhance transcription of that particular gene, okay? And again, their sequence, so there are regulatory proteins that will bind to that enhancer, right? And that will control it, okay? So this, again, enhancer, enhance, increase transcription. Okay. So that's what I just was saying here. So an activity of any given enhancer is specific for the promoter of its target gene. They're not general, they are specific, just like we gave the example of those immunoglobulin genes, right? They're very, very specific. Okay, and so there actually is another type of protein that sort of helps maintain this specificity. And so something called insulators will actually sort of divide the chromosomes into independent domains, so that certain enhancers won't be able to act on other promoters that might be near it, right? And so that's just sort of a kind of a side note there to sort of help with understanding how these can stay specific. Okay. So that's what this is sort of showing you here. So this is, it's, so because of those, I don't even have this on the review sheet, right? I'm not really going to ask you about this too much, but this sort of was setting up the idea of these insulators that sort of divide up and make different domains, right? This is a specific example of that. Okay, so you can kind of describe genomes as being divided up into something called the topologically associated domain. And basically enhancers and promoters that are within a certain domain right? They're the ones who will interact with each other a lot, but not with elements in other domains. Okay, so again, this is a way of sort of organizing it, all right? So it says the main protein that binds insulators, okay, invertebrates is called CTCF, all right? So you can see here, right, that this is going to bind to these insulators. So you can kind of see here, this is showing you sort of boundary between these domains here, okay? Again, you don't need to worry about it, right? So this is kind of showing you here, this is sort of one topologically associating domain. And so that everything here, right, things will interact with each other, right? So this enhancer is interacting with this other types of, with this promoter here, and essentially the transcription machinery that's going to transcribe these genes here. And again, because you sort of have this looping that occurs, and I kind of said that before. So this is just getting into a little more detail. Like, again, you don't need to know the specifics here, okay? But again, just another sort of level of kind of understanding how these things actually work within the cell, right? And some of the mechanisms involved. Okay, in terms of how we have identified these transcription factor binding sites, right? There's lots of different ways that we've identified them, and here's an sort of example. And again, I'm not really going to ask you about this on the exam. Okay, so there's this mobility shift assay where basically you can radio label DNA fragments, right? And then they get incubated with a protein and then analyzed. So bottom line is if that DNA sequence or that fragment that you have can bind a protein, meaning it's a transcription factor binding site, right? So basically you have these different fragments, right? You add transcription factor to it. The transcription factor will only bind to those fragments where it has that sequence that it would normally bind, right? And those that have those fragments of DNA that actually have protein bound will move much slower in the gel. Okay, so that's what this is showing you here. So it is a way of identifying, so then you know, all right, well, I know if this protein, if this fragment of DNA is bound by protein, I know that the sequence where that protein binds is within this fragment. And so then you can just basically cleave this out and analyze that sequence and figure out what that sequence is and figure out that DNA binding site. Okay, so if you see that now, so it's showing you here though that these were radio labeled, right? And that's the way you're visualizing it. Okay. Okay, another type of method that basically allowed us to identify transcription factors. Okay. All right, so you can, again, I'm not going to ask you guys about this on the exam. All right, so you can see here, you can treat cells with formaldehyde that will basically cross-link those transcription factors bound to the DNA, and then you basically can fragment these, and then you can identify, right, where these are bound. Okay, so then you can have a specific antibody that will recognize that protein and pull out that one that you're interested in. Okay, so these are all ways of identifying and studying these transcription factors and also sequences that they bind to in the DNA. So remember, a transcription factor in general, and you should write this down somewhere on your review sheet there, a transcription factor is a protein that binds to DNA. Okay, that's what it is. We've talked about different types already. We talked about general transcription factors that are involved in just basically initiation of transcription, and then we're starting to talk about some specific transcription factors. Okay, or we're just kind of talking about we haven't given examples of them yet. All right, so anyway, this is continuing here. You don't need to know these specifics, but if you're interested, there's a little bit more details here, okay? But you can basically identify this, right? We're talking about identifying that transcription factor binding site. These are proteins that bind to DNA. This is the binding site, DNA, a specific DNA sequence, and this is the protein. Okay, so one of the first transcription factors that was isolated was something called SP1, okay? And it was basically using that SV40 virus that we talked about before. And this SP1 was shown to bind specifically GC boxes, okay, in that particular promoter. So remember a couple slides ago, I showed you that SV40 promoter, and I didn't really say much more about it. Okay, and this is kind of important to to know, because this was one of the first transcription factors that was isolated. Okay. All right. So it says once they basically were able to see that it bound to the promoter, then they sort of knew what the function of SP1 was. Okay. And so this is actually sometimes I give you guys actually this key experiment to read. And so that's why this is pulled from that. But in fact, you don't have it to read. I'm not even going to bother pulling it up. I can, because I think I have the PDF on Moodle. So if you're interested, you can read that. But let me just see if I can. Yeah. So basically, this is just showing you, and so he used a different technique in terms of identifying this and pulling it out. So I gave you a little bit of example of some other sort of older type of techniques. Here, he used this DNA affinity chromatography. Okay, and so we can go back to that picture in a second. So this is basically showing you one of the main figures from that paper, right, where he basically was able to identify these. So yeah. So basically he's able to sort of pull out that SP1 here. Okay. So this is, again, it's showing you a gel, right? You can see those various bands. And so this is just sort of initially all the different proteins that you get, but then he's able to purify and pull out just the transcription factor. Okay. So a little more on that. And so he used this DNA affinity chromatography. Okay. So again, the specifics to that method are not on the exam as well, but I like to introduce some of these methods. So, so basically you can bind, so it's kind of the same idea that we've been talking about before, right? So it's, you take pieces of the DNA, right? So here it's saying you basically can take, pieces of DNA, right, with repeat GC box sequences that you believe are going to bind your transcription factor, right? And then basically pass cell extract through it. And the SP1, that transcription factor, will bind to all those places where, the GC box is, okay? And then, like I said, you can purify it from there, okay? So this is a little more sophisticated method, right? So it's using these agarose beads to basically catch that SP1 protein, okay? So see it here in the column. Here's your beads, agarose beads with the DNA sequence, right? That's going to match this SP1 that's found here. And then the SP1, any SP1 that's in the cell extract will bind to this GC box sequence of the DNA. And then you're able to basically capture just those, let all the other proteins flow through, and then eventually just elute off your SP1. So it's a way of purifying it. Once you purify that SP1, then you can study it further, right? And so it's really important to have techniques like this to be able to isolate these proteins. Okay, so transcriptional activators like SP1 will bind to regulatory DNA sequences, right? And they will stimulate transcription. And they, in general, have sort of two different domains. One domain that will bind DNA, the one that will stimulate transcription. Okay, so we talked about enhancers. They will enhance transcription, right? Transcriptional activators, again, this is an example of a transcription factor like SP1, will bind other regulatory DNA sequences and basically activate transcription. So an enhancer sequence isn't the only sequence to which a transcription factor can bind, okay? And in fact, it's not. There's many, many regulatory sequences in the DNA. All right, so this is showing you here. So here's an example of a transcription factor. And if we want to be specific, we can say SP1 that we just talked about in length, right? SP1 here has two different domains. It has a DNA binding domain. And so again, think for a second why that's important. It has to be able to bind to its regulatory sequence in the right spot, right? And then this activation domain will interact with all these general transcription factors that we talked about before to basically stimulate and activate transcription, okay? So remember, this is regulation of transcription. All right, and again, if you're on your review sheet here, it says DNA binding domains. So we're going to talk about a few different DNA binding domains, and they're here, okay? So you just need to know examples of these, okay? I don't ask a million questions about it, but we should know the different types of DNA binding domains that exist, okay? So it says, many transcription factors have now been identified in eukaryotic cells. We just gave one example of that SP1. That was one of the first ones that have been identified, but using techniques like we talked about, right? That affinity, purification, that affinity chromatography, using techniques like that, we're able to basically identify and isolate many, many different transcription factors. So right now, about 2,000 have been identified, okay? All right, so anyway, many of them have these sort of 4 distinct types of DNA binding domains, okay? So zinc finger, helix turn helix, leucine zipper, and helix loop helix. Just know these examples, write them down on your review sheet, okay? And that's all. We're going to leave it at that. You can talk about these in a little more detail, but we're not. So remember, that's the DNA binding domain, binds the DNA. There are 4 different types we just mentioned. Activation domains, we don't know as much about. Okay, we know that they interact with the general transcription factors and the mediator proteins, and basically stimulate transcription. Okay, that's what you need to know. We also know that they will interact with other activators that will help modify chromatin structure. So bottom line is, though, those activation domains will interact with whatever proteins they need to interact with to stimulate transcription of that gene. Okay, so that's what this is showing you here from this picture, just what I said. Okay, and again, remember in the DNA binding domain, there are different types of DNA binding domains. Okay, so you don't always turn on genes, right? Or you don't always increase transcription. You also can repress it, okay, which will inhibit. And so sometimes they'll simply interfere with the binding of other transcription factors. They can also compete with activators. Okay, so it sort of depends on what the mechanism is. All right, so again, include interactions with mediator proteins, general transcription factors, or other specific activator proteins. They can really interact with anything, but the bottom line is a repressor is going to turn down transcription or inhibit transcription. Okay, so here showing you, right? So this is some activator, some transcription factor that would normally activate transcription, except now there's a repressor bound, and therefore it can't bind, okay? Therefore, transcription will not be initiated. Here, again, just like we kind of showed with the activator, it's the same idea. It has a DNA binding domain, this repression domain. A repression domain will interact with all those general transcription factors to basically stop transcription. Okay, so that's all regulation at initiation, and most of the time, that's when it's going to happen, okay? If you look at your Chapter 8 homework, it says, how do enhancers differ from promoters as cis-acting regulatory sequences in eukaryotes? I mean, enhancers can be, right? Remember, they can be really far away from the transcription start site. They can be downstream, right? They can be inserted backwards. And so they don't need to be right in front of that transcription initiation site. And they will then interact with those general transcription factors that are binding or that are bound at the promoter, okay? And so they're different in those ways. All right, so continue on here. But you can also regulate transcription at elongation, but again, a lot of it happens like we were talking about at initiation, okay? And so again, if you're following along your review sheet, we're now at regulation of elongation. Yeah. Okay, so there have been studies that have shown that molecules of RNA polymerase can actually start transcription, but then are stalled. Okay, so just so you know, that's an option. We don't know as much about that. I don't think there's many examples of that. Most regulation of transcription is going to occur at initiation. The cell does not want to start transcription if it's not going to finish it, right? That would be a waste of resources. Okay. So again, this next part is chromatin and epigenetics. I said if we're going to talk about regulation of transcription, right, a lot of that has to do with modification of the chromatin, okay? So, and again, we talked about how DNA is packaged, right, with histones, that basically the DNA is wound around. You can modify those histones. The cell can modify those histones to actually make that DNA more available for transcription, okay? And we said, it says many histone modifications are inherited when cells divide. That's the epigenetic part of it, right? We've already introduced this concept before. Okay, so one of the ways that histones can be modified is something called histone acetylation. And I told you, you will read a paper after you have this exam on histone acetylation in its relationship with Parkinson's disease, okay? And so here, acetyl groups are added by a specific enzyme called histone acetyltransferase, HAT, and they're actually removed by HDAC, histone deacetylase, okay? So we should know these enzymes here. And what happens if you acetylate, or if you have this acetyl group that's added, right, by HAT, it will neutralize the positive charge of lysine in the histone and will relax the chromatin structure. And that will lead to increased availability of the DNA for transcription. On the flip side, if it's deacetylated, that acetylation is removed, it's going to be more tightly wound and transcriptionally inactive. Okay? There are many transcriptional activators and repressors that will associate with both the histone acetyltransferase and the HDAC to basically regulate transcription. Okay, so it's not only about talking about transcription factors and where they bind on the DNA, but they also a lot of times will be interacting with the histones, right? Or with these enzymes that will affect the histones that can make that DNA more available. Okay, so again, these are epigenetic regulations, meaning the histones that's modified then can get passed down to the next generation. Okay, so that's what this is, basically showing you here. You can see this sort of acetylation that's occurring, and it's particularly going to be occurring on a lysine. Okay, that's where the acetyl groups are added, right, to those histones. Remember, histones are proteins made up of amino acids, particularly if you're going to talk about, these two enzymes. HAT will add the acetyl group again to a lysine. HDAC will remove. You should know this. Okay, so this is again showing you here where you have a co-activator, activator that here's your activator, and you can see that it's associated with HAT, with that enzyme, right? Or that this HAT, which will add acetyl group and will help relax that the packaging, right, and make that DNA more available, right? A lot of these activators are also associated with that enzyme, okay? And vice versa, if you talk about a repressor, a lot of times they're going to interact with HDAC because that's going to make that region of DNA transcriptionally inactive, okay? So we should know the difference between HAT and HDAC and what it does, right? So HAT adds an acetyl group, and when it adds an acetyl group, what happens here? The positive charge is neutralized in lysine, and it relaxes the chromatin structure. So again, remember that DNA is negatively charged. So if you're reducing the positive charge of the histone, it's going to, basically reduce that interaction between that positive and negative charge because you're essentially making the histone less positive, okay? HDAC will remove an acetyl group. Okay, we saw that here, and therefore it makes sense that repressors will actually interact with HDAC. It makes sense that activators, or transcription factors that are going to activate transcription, interact, in fact, with HAT. Okay? Okay. You can also modify histones not only by adding acetyl groups, okay, but also methylation and phosphorylation as well. Okay, so it says methylation of lysine and arginine, phosphorylation of serine. And so also additions of other small peptides to the histones can also modify them. And then again, it's going to have the same effects, right? It's either going to make the DNA more available or make it less available. It's either going to inhibit transcription or enhance. Okay. All right, so it says histone modifications affect gene expression by altering chromatin properties and by providing binding sites for proteins that activate or repress transcription. Okay, so it says here methylated H3 lysine and lysine 9 and 27 residues are binding sites for proteins that induce chromatin condensation and formation of heterochromatin. Okay, so this is this particular part of histone 3, right, that gets methylated and it's involved in the formation of heterochromatin. Bottom line is, understand that these histones are often modified, right, or and not often, I mean, pretty much all the time they're modified. And depending on how they are modified, that's going to affect how they interact with the DNA. Okay. And again, it will either make that DNA more available for transcription or not. Okay. So the big one that we should understand all the details for are acetylation. Okay. And like I said, we'll have a little follow-up paper that kind of talks about why we should care about this. Okay. This is showing you kind of everything, right? So here's histone. It's showing you this N-terminal tail of a histone protein, showing you can have some methylation, right? Or acetylation on the lysines, showing you phosphorylation here on serine, right? And also, that's all of them, right? So the three ways in which we're kind of showing here that these histones are modified are phosphorylation, acetylation, and methylation, okay? And so basically, if you're removing some of these, then that chromatin is going to be more inactive, right? By adding acetyl groups, remember we said that those are going to lead to more active chromatin or more active transcription, okay? All right, so this is actually, because again, we're talking about chromatin and how this is sort of is involved. Promoters and enhancers, like we talked about, they have really distinct chromatin features. They actually don't have nucleosomes there, and they're basically always accessible for transcription factors to bind, okay? And so you can actually identify these by adding DNase to digest them. And so essentially, the DNase will cleave these areas where, right, there are no histones because it's basically naked DNA. That's what this is showing you here. Makes sense, right? Okay, again, we talk about epigenetics already and what epigenetics is, okay? Modified histones, like we just talked about, is a really big part of epigenetics, okay? They're transferred to both progeny chromosomes, right, where they basically will direct similar modification, meaning, you know, one histone from the parent cell is going to direct that next, so say it had, I don't know, whatever, 2 acetyl groups added onto 2 of the lysines. It's important that then that daughter, the histones in the daughter cells actually have that same modification, okay? So you're maintaining that same pattern. All right, so that's what this is kind of showing you here, that you want to maintain the same modifications, right, from the parental chromatin to the progeny. Okay, so I showed you a picture of this already. I mean, again, there's not really, I mean, this is kind of just showing you that these modified histones are sort of directing, these modified histones, when the new histones are inserted, they're basically inducing them to have the same modifications as they did. And that's what this is showing you here, okay? I'm not going to, you're not going to need to explain in detail what's going on, but it's pretty clear to see. In fact, I guess this picture is probably the best one to look at that, right? Newly incorporated histones, you can see that they're not modified, but then these guys will direct the modifications so that you end up with these having the same exact modifications as their neighbors, essentially, okay? All right, that was all talking about histones, so don't be confused now when I just mentioned DNA methylation, okay? That was all histones. So we talked about histones. The one main thing we talked about, again, we probably want to make sure we write this in, was acetylation. You guys have that there. So where acetylation is on your review sheet, you should be writing HAT and HDAC and write what HAT and HDAC do, okay? You can also phosphorylate and methylate histones as well, and that will affect or modify those histones and make transcription either more active or less active. However, you can also methylate DNA, okay, which is another sort of epigenetic sort of mechanism, okay? And so basically this is adding a methyl group directly to the DNA, okay? And that the methylation or the addition of these methyl groups is actually correlated with repression of transcription, okay? So a way of controlling. So it says it's a common in transposable elements. Remember we talked about those in chapter 6? That these are pieces of DNA that can actually move around. And so if those regions, those transposable elements are methylated, it will suppress their movement, okay? Also involved in repression of mammalian genes, okay, that are involved in particularly in development. And so it gives you an example, or it's written on your review sheet about genomic imprinting, okay? And so that's here, okay? And so we'll talk a little bit about that in a second. So again, I think I already said this, now it's in bold, right? DNA methylation is an important mechanism for epigenetic inheritance. So if I asked you to give me examples of sort of things involved in epigenetic inheritance, we talked about histone modification, but also DNA methylation is one as well, okay? And we also did talk about this a little bit when we were talking about replication, right? So it says following DNA replication, an enzyme will actually methylate sequences of the daughter strand, okay? And so that's what this is showing you here. Here's your methylated parental DNA. Here's DNA replication that occur, and eventually those daughter DNAs will get methylated as well. Okay, but remember, initially they're not, and that was one of the ways that the cell can identify what was the newly synthesized versus the parental strand, and could actually use it to help with repair. But then eventually it does get methylated. Okay, so this does play a role in genomic imprinting, right? So it says expression of some genes depends on whether they come from the mother or the father, okay? So for example, there's this gene H19 is transcribed only from the maternal copy. It's methylated during development of the male, but not during the female because, so if it's methylated during development of the male, it's not going to be expressed. But in the female, you want it to be expressed, okay? And so that's what this is showing you here. Right? So if you're talking about your gametes, right? So sperm cell versus the egg cell, here's this H19. You can see in the sperm cell, it's actually methylated, which keeps it inactive. And here it's active. We also talked about, remember some of those different types of RNAs involved in X chromosome inactivation. So that's a different mechanism, but kind of having the same effect here, right? So again, you only want this gene to be active in the maternal copy of the chromosome. Okay, and so therefore, it's actually methylated in the paternal. Okay, so that's the genomic imprinting example there. Okay, last thing to talk about is RNA processing and turnover. So it says bacterial mRNAs are used immediately for protein synthesis, so they don't need to be processed. Most RNAs, however, do need to be processed. Okay, and so it says regulation of processing actually provides another level of control of gene. Okay, so ribosomal RNAs are derived from this long pre-ribosomal RNA, and then they actually have to be cleaved, okay, to form those three different RNAs like that we've sort of been talking about, right? And again, we'll talk a little bit more about the 16S, 23S, and 5S, okay? So again, it starts off as this really long pre-RNA, pre-rRNA, and then they get cleaved up into these different forms. Eukaryotes actually have 4 types of ribosomal RNAs, and then 5S is actually transcribed from a separate gene, okay? So in prokaryotes, you form these 3 ribosomal RNAs. In eukaryotes, you have 4, but the 5S, again, is synthesized from a separate gene, okay? So you can see here, right? This is the pre-ribosomal RNA, this really long transcript here, and then it gets cleaved. So this is processing, right? It gets cleaved to form these. And then eventually gets, processed even further to form those mature ribosomal RNAs. You can see here in eukaryotes, right? Here they are. Here 18S, 5.8S, 28S, and then remember the 5S is going to come from a different gene, okay? Okay, so that's for ribosomal RNAs. Their processing is that they start off as a long pre-rRNA and they get cleaved, okay? For tRNAs, they actually also have to be processed as well, okay? So they also start as long precursors, and then they get cleaved. Okay, so processing the 5' end of a pre-tRNA involves cleavage by the enzyme called RNase P. And again, it's a ribozyme. Okay, so it basically is an RNA. It's not a protein, so it's an enzyme that's an RNA enzyme. And then there's also processing that occurs on the 3' end, and basically it adds this CCA terminus. Okay, and that's actually where the amino acid will attach. Also, in tRNA, bases get modified, okay? So those are there's a couple ways in which the tRNA is processed. So first, right, it's longer, so this RNase ribozyme will cleave this part off, and then you end up with this, and then you're going to have several modifications of the 3' and the 5' end that we talked about, right? So you have a CCA that's added to the 3' end, and then you also have modification of bases. Okay, so these sort of normal bases that are here get changed, okay? And that allows some of this base modification allows for, and we'll talk about this later, but something called, remember how we talked about wobble, right? If you talk about the codon and that at that third position, it could really bind to a couple different nucleotides via complementary base pairing. It's because it has these modified bases. Okay, so this is giving you some examples here of these modified. Okay. Also, some pre-tRNAs have introns that actually have to be removed by splicing. So the transfer RNAs have to be modified quite a bit before they're ready to actually function, okay? And so I have some of these listed here. For tRNA, cleavage at the 5' end, right, by that ribozyme, addition of CCA at the 3' end, and then also modification of bases. So you should know those. You need to know the specific details in terms of how all those are happening, no. But if I ask you how tRNA is modified, we should be able to list those ways. Okay, so for mRNA, this is the last, basically almost the last topic here. The pre-mRNAs are extensively modified before they can be exported from the nucleus, okay? It says transcription and processing are essentially kind of coupled, but remember, transcription is happening in the nucleus, then that pre-mRNA has to be modified, and then it can be transported out of the nucleus to go find the ribosome and be translated, okay? It says the C-terminal domain of RNA polymerase II will actually play a key role by serving as a binding site for enzymes that are involved in processing. So sort of as that transcription of that mRNA is happening, or kind of as it's ending, you start to have that mRNA being processed. Okay, so that's what this is saying there. Okay, and so how is it processed, right? That mRNA, and we probably mentioned these before, I mentioned this in Bio 111, here's your pre-mRNA. That's formed. You have a 5' cap, you have a 3' poly A tail, and you have introns spliced out. Okay, super simple. You need to know those three things. Okay, 5' cap, a little more detail on it. Basically, it is modified by the addition of a 7-methylguanosine cap. Okay, what is the function of that 5' cap? And you probably should write this in your review sheet, is that it will stabilize the RNA. It will also help align it on the ribosome during translation. That 3' poly A tail, right? All right, this is just giving you a little bit about the mechanism. You don't have to worry about that. Recognition of that poly A tail leads to termination of transcription, right? Cleavage and polyadenylation of the mRNA. So sorry, this is all about mechanism here too, right? That there's this polyadenylation signal that essentially terminates transcription, and then you have the addition of that poly A tail there. Yeah, so any RNA that's been synthesized downstream of that poly A tail is actually degraded, okay? And so that's what this is basically showing here, in terms of the mechanism. I'm not going to ask you about the mechanism in terms of how that poly A tail is actually added, okay? Just know that the poly A tail is there, okay? And that, again, it sort of helps to signal that that's the end of that transcript, right? And so anything past it will be degraded. It also serves as sort of protection after the fact, as sort of protection from degradation, okay? All right, number 3, introns are spliced out. We talked about this many times, okay? We should know what that means. And so those introns have to be spliced out because they are not coding regions of that RNA, of that mRNA. Because again, we're talking about mRNA is going to go and get translated. Okay, and we mentioned this before in terms of alternative splicing, okay? So again, that involves those introns, right? It says most pre-mRNAs have multiple introns, and so different mRNAs can be produced from that same gene. And again, this is a way of controlling gene expression, right? And also increases the amount or the diversity of proteins that can be encoded from one transcript, essentially, okay? All right, so it says over 90% of pre-mRNA sequences are introns, okay? We even talked about this in chapter 6 when we're just talking about DNA in general, that there's lots and lots of non-coding sequence. Okay, processed mRNAs are protected by capping and poly A, okay? But the unprotected enzymes introns really get recognized and degraded by enzymes, okay? And so once those are spliced out, those introns, they just quickly get degraded because they have no protection, okay? All right, Also, if you have a defective mRNA, it can also be degraded. Okay, and so this says this is some mechanism here that the cell has. And so now we're sort of under RNA degradation, right? So again, remember that mRNA, the presence of the 5' cap and the poly A tail really protect the mRNA transcript. It needs to be translated from degradation. But if we talk about cleaving out introns, where do they then go? They just get degraded. Also, if you have an mRNA that is defective in some way, it will also be degraded, okay? So it's degraded rather quickly unless it has these sort of protective mechanisms or modifications on them. Okay. All right, so it says, so this nonsense-mediated mRNA decay, right? It'll eliminate any mRNAs that lack complete open reading frames, right? Meaning that they're missing codons essentially. And so if the ribosome will encounter a premature termination codon, it will stop, and then the mRNA will be degraded. Okay, and again, because of sort of this innate sort of I guess sort of feature of the RNA, that it is pretty unstable. Okay, ultimately RNAs will be degraded. Okay, and so this is just sort of another level of regulation. So even if it does have the 5' cap and the 3' poly A tail, it will only survive for a certain amount of time in the cytoplasm. It's not going to hang out there forever, right? Because then you would lose a level of regulation, right? It's there for a certain amount of time, gets translated, and then if the cell still needs more of it, it's going to have to make more. Okay, it can't just hang out there forever. The cell would have no way of controlling how much it would need. Okay, so it says levels of any RNA are determined by balance between synthesis and degradation. Okay, and so rates of degradation, like I just said, can kind of control gene expression. And so if you look at the review sheet, it says, how does this affect gene expression? Write it in there. Write what you think that answer is, and essentially it's here. Okay. Also, if you go back to your Chapter 8 homework, 3 ways in which pre-mRNA is processed. I hope that's easy to answer that question, right? The 5' cap, the 3' poly A tail, and splicing. Introns are spliced out, okay? So it says our rRNAs and tRNAs are very stable, okay? So, and more stable than mRNAs. All right. So it says this accounts for the high levels of these RNAs in the cells. But mRNAs are relatively unstable, and for very good reason, because this can affect gene expression. So, for instance, there's differences between stabilities of just mRNAs in general, right? So if you have a mRNA that is sort of in general transcribed a lot or translated a lot, maybe that one's a little more stable than something that doesn't need to be made all the time, okay? All right, so in terms of bacterial mRNAs, they're rapidly degraded, okay, super quickly. Half-life of about 2 to 3 minutes because they don't have as much regulatory capacity as eukaryotic cells do, okay? And eukaryotes, mRNA, like I was just saying, half-lives will vary. Some can be 30 minutes, some can be up to 20 hours, okay? This is what I was just saying. Short-lived mRNAs code for regulatory proteins, okay? They need to vary or change quickly, okay? mRNAs that encode for structural proteins, right? Or metabolic enzymes that are used all the time in the cell, they have really long half-lives. Okay, so again, if you look at that last part of the review sheet, how does this affect gene expression? You want to, add in some of these things we just talked about. Okay, so RNA degradation is also involved in regulating gene expression. Okay, all right, so this is a mechanism in terms of how that mRNA is basically degraded. Okay, so shortening of that poly A tail. So and then you basically have nucleases that will chew it up. You don't need to know this process. All right, and finally, that's the end. So we went over the homework questions. I was drawing your attention to that review sheet. You need to make sure it's completely filled in and you are submitting it as an assignment. All right, thanks.